Good morning, class. Welcome to the third session of class 10th biology. Today, we are going to start with the second chapter, that is structure of chromosome. As you can see here, chromosome. First of all, I should tell you about what are we going to study in this chapter. So we will be studying the structure of chromosome. We will be studying the basic structure of chromosome with elementary understanding of terms, those terms such as chromatid, gene, structure of DNA, and centromere. In the previous class already we have studied about cell division, the type of cell division that takes place, and what are its utility and function. So we all know the genetic material which is present in the nucleus is in the resting phase. In the resting nucleus, that is a non-dividing nucleus, is in the form of a network formed with of long extremely thin fibers. These thin fibers are nothing but the chromatin fibers. So what are chromosomes? Chromosomes are small rod-shaped darkly stained bodies visible only during metaphase and anaphase of nuclear division. In the last class, you know very well, I had explained you about the various phases, that is metaphase and anaphase. So during this phase itself, we will find these rod-shaped darkly stained bodies. Chromosomes are the genetic material in the resting phase in the form of a network. It forms a network inside the nucleus. They are composed of long, extremely thin fibers, and these fibers are called as chromatin fibers, and they form a net-like structure, and which is termed as the chromatin net. When a cell enters the division, the chromatin fiber starts to condense. In the previous class itself, I had told you, when I had explained you about cell division, that when cell division starts, there are a lot of changes that takes place in the chromatin material. So what actually happens? The chromatin fiber starts to condense. Not only it condenses, it attains into thick, darkly stained bodies, and these darkly stained bodies are called chromosomes. Chromosomes are formed of nucleoproteins. What are the things of which these chromosomes are formed of? So chromosomes are formed of nucleoprotein. What is this nucleoprotein? Nucleoprotein means nucleic acid, which is DNA, and basic protein, which is in the form of histones. These basic proteins are histones. Chromosomes are called as the genetic vehicles. Genetic vehicles of genetic are called as the vehicles of genetic material. Why are they called as the vehicles of genetic material? Because they transfer the phenotypic characteristics from one generation to the other generation, means from the parents to their offspring and so on. It is called as a genetic material as they are transferred from parents to offspring during the cell division. Then when was this chromosomes discovered? So discovery of chromosome is attributed to, it was first of all observed by Hofmeister, a scientist, in the year 1848. Where did he see? Where did he found the chromosomes for the first time? He found the chromosome in the nuclei pollen mother cell of a plant called Tradescantia. Have you all seen a Tradescantia plant? I will show you what is a Tradescantia plant and how does it looks like. This is a Tradescantia plant. It is a very common plant. You will find it growing in the as an ornamental plant in your garden, in various botanical gardens also you can find. In our school also you will find it is present there, and we are using this plant for various practical purposes. You will be definitely using this in your higher classes when you will go. We will be using this for various kind of practical. So this is a Tradescantia plant. So I have told you that discovery of chromosome, it was first of all observed by Hofmeister. You have to keep these dates and the name of the scientist in mind because it is asked during board examination. So in the year 1848, the term chromosome was for the first time used by Welder in the year 1888. Now let us move on to the next topic, that is chromosome number. Chromosome number, every organism, every species has got a particular chromosomal number. This number is constant in all the cells of an individual and individuals of a particular species. Means in a particular species, all the individual will have the same chromosomal number, but individuals belonging to different species will have different chromosomal number. Therefore, number of chromosome is helpful in identification of species. Once more I'll tell you. Therefore, number of chromosome is helpful, means when we observe the number of chromosome, it is a point of identification, or it is helpful in identifying a particular species. Some of the examples of number of chromosomes in plants and animal cell, we will be seeing some of the examples. Like, see, around us there are so many plants and animals. So out of them, some of the examples I have chosen. The first one is onion. Onion, the botanical name is Allium cepa. The number of chromosome, that is in diploid number of chromosome is 16. Next one is rice. That is the botanical name is Oryza sativa. The diploid number of chromosome present in rice is 24. Wheat. The botanical name of wheat is Triticum vulgare. And the diploid number of chromosomal number which is present here is 42. Next one is very common, that is us, that is man, Homo sapiens. And the chromosome number in a diploid form is 46. The next example we'll be seeing cat, that is Felis domesticus. And the diploid number of chromosome is 38. Let us see one more example, that is tiger, that is Panthera tigris is the zoological name, or the scientific name is Panthera tigris. It has also the same chromosomal number like that of a cat. That is why we find too much similarity between a cat and a tiger. Now we will be moving to the next topic, that is diploid and haploid number of chromosome. You already know about diploid and haploid number of chromosome, which was explained in the last class. So we will be moving further with the same explanation. First of all, we will see what is a diploid number of chromosome. As we all know, each cell of body has got two sets of chromosome. In our whole body, the cells which are present, they are having pair of or two set of chromosomes. Here, one set comes from the father side through the sperm cells or through the male gamete, and the other set comes from the mother side through the ovum or through the female gamete. Then what happens upon the fusion? What happens? It is represented by, it forms a diploid form. A diploid number of chromosome is formed, and it is represented by 2n. These are the two ways how it can be represented. It can be represented by 2 small n or 2 capital N. These are the two ways in which this diploid number can be represented. Next is haploid number. So what is haploid number? Each cell, when we are talking about basically usually in the germ cells or gametes of diploid organisms. This was also explained to you in the last class, that the gametic cells or the germ cells, they are When they produce gametes, they are haploid in number, means they are not found in pairs. They contain, the germ cells basically, they contain only one set of chromosome. And when they contain only one set of chromosome, they are represented by small n or n. So whenever you will find any chromosomal number explained as 2n, it means it's a diploid number. Whenever you will find it written in the form of n or capital N single, then it is a haploid number. We will be moving on to the next topic of today, that is structure of chromosome. We can see here the basic structure of chromosome. Each chromosome appears in its condensed form during the start of cell division. When cell division starts, the chromosomes become condensed. It consists of two chromatids. You can see here the two chromatids. These are the two chromatids. You can see here I have made two diagrams. Actually, this is a chromosome with two chromatids. You can see here two chromatids. And here you can see a chromosome with one chromatid. This is only a one chromatid. This only will, upon duplication, will develop into two chromatids. It consists of two chromatids joined at some narrow constricted portion. Can you say this narrow constricted portion? Okay. So this narrow constricted portion is termed as centromere. This is marked as centromere. You know what is the other name of centromere? It is also termed as primary constriction region, or also by the name kinetochore. The region of it is the same place through kinetochore only, that it is a point of attachment between the chromatids and the spindle fibers during the cell divisional process, as it was explained in the previous class. The region of chromosome on either side of the centromere is called arm. You can see here the either side. This is the centromere. On either side of the chromosome, you can find the two arms. If you will see, you will see one arm is short, whereas the other one is long. So it is termed as the short arm and the long arm. Okay. And you will find this green color coil structure. This is the DNA, which is in the form of coil wrapping around this chromatids. Next, centromere. Centromere, this is the constricted part in the chromatids. It is also the point of attachment. So chromatid is the point where spindle fiber are attached during cell division. One more time I will tell you. It is a point, means centromere is a point where spindle fibers are attached during cell division. The actual point where this connection takes place is called as the kinetochore or centromere. What is the function of this centromere? Centromere, it determines the shape of the chromosomes. So the shape of the chromosome is determined by this centromere itself. It provides attachment for spindle fibers. Just now I had told you about the attachment. So this is one of the very important function of centromere, that it provides attachment for spindle fibers. It also helps in the movement of chromosome during cell division. The sister chromatids, they get separated at the equator and start to move towards their respective poles in the anaphase, isn't it? So how does this movement take place? As explained earlier, it takes place by the constriction or by the contraction of spindle fibers. So this centromere will form the attachment to the spindle fiber, and therefore it will be helpful in the movement of chromosome during cell division. We are going to start with the topic moving ahead, that is types of chromosomes. So the different types of chromosomes we are going to see. Chromosomes are classified based on the position of centromere. So I had told you centromere plays a very important role. In the function of centromere, already I had explained you that centromere, the first function that I had told you was it determines the shape of chromosome. So depending on that itself, we will be seeing on what are the different shape of chromosomes. So the first one is telocentric, second one is acrocentric, then the third one is metacentric, and the fourth one is submetacentric, and the fourth one is metacentric. So what is telocentric? Telocentric are rod-shaped chromosomes with centromere occupying a terminal position. So centromere you will find on one end. At one end you will find this centromere located. It will be rod shaped with terminal end of centromere. So rod shaped chromosome occupying the terminal position. They possess only one arm. Next one is acrocentric. These are also rod shaped chromosome, but here the centromere is subterminal. Okay, means it will not be situated at the end point, but just after that a small arm will be present. Means one arm will be very long, whereas the other arm will be extremely short. Rod shaped chromosome with subterminal centromere. One arm is very short, whereas the other one is very long. Let us see the submetacentric. The next one is submetacentric. What happens in submetacentric? These submetacentric chromosomes, they attain an L shape. L shape. Okay. They are also called as L-shaped chromosomes, where the centromere is slightly away from the midpoint, and two arms are unequal. You will find it is not in the midpoint, but it is slightly away from the midpoint, slight away from the midpoint, and it will form an L alphabet, and the two arms will be unequal. Let us come to the next one, that is metacentric. Metacentric chromosomes are also termed as V-shaped chromosomes because they attain a V English alphabet V. The shape is like that. Here the centromere will lie in the center. It will be lying in the midpoint of the chromosome, in the middle, with two equal arms. It will be situated in the midpoint, and we will find both the arms of equal length. So this was all about the types of chromosome. This is also very important. I will be explaining you again this thing through the PowerPoint. Fine structure of chromosome. The next thing, that is fine structure of chromosome. We know chromatin material condenses to form chromosomes. Already as it has been told. Chromatin is formed of two substances. So what are the two substances of this chromatin material or the chromosome, which is of which it is formed of? So the very first thing, the first thing of which it is formed of is deoxyribonucleic acid or DNA. Whereas the second thing of which it is formed is the basic protein, that is histone proteins. Now coming to the composition of this chromosome. DNA forms 40% of the chromatin material. If we take the whole chromatin material as 100%, then 40% is comprised. The DNA comprises of 40% of the total chromosome or the chromatin material, and the remaining 60% is made up of the histone proteins. So 40:60% is the ratio between DNA and histones. DNA and histone, they form a complex structure. What kind of structure will they form? Actually, these histones, they are 8 in number, 8 molecules of histones. Histone forms small core and it will form a centrally small core particle, and the DNA strand is found wrapped around it. It forms a structure called nucleosome. 8 molecules of histones wrapped by a DNA strand, double-stranded DNA strand will form the structure, and that structure is called as nucleosome. You can see here the structure of a nucleosome which I have drawn. This is a typical structure of a nucleosome, how it will look like. We will find here histone proteins which are represented by red circles. If you count 1 2 3 4 5 6 7 8, okay, 4 at the back, 4 in the, so 8 molecules of histones you can see here, and you can find a DNA which is forming 2.5 loop around this core histone protein, and you will find it is joined by a linker histone which keeps intact the DNA. It will not allow the DNA to move from its place. So this is the function of the linker histone. Histone form the structure of called the nucleosome. Each nucleosome has one core particle formed of 8 molecules of histones. DNA makes 2.5 coils around it. I have told you this DNA strand, it will form 2.5 coil. It will wrap around this core particle in 2.5 coils. Thus, chromatin fiber has numerous, when we are talking, so like this, so many nucleosomes are present. Chromatin fiber has numerous nucleosomes which are arranged like beads on a string. Beads on a string, or you can also imagine the structure of pearl necklace. Okay, in the same way like pearl necklace, these are the beads and this is the string. So it can be also observed in that form. Structure of DNA. DNA is this DNA when we are talking about, DNA is a macromolecule. It's a big molecule. DNA is a macromolecule, and it is formed of 2 polynucleotide strands. These strands are complementary and anti-parallel to each other. When we are talking about the strands of DNA, these are the ends of the DNA is represented by 5 prime and 3 prime. Here you will find that the 2 strands are anti-parallel to each other. If it means if one strand is starting from 5 prime to 3 prime, the complementary strand will start from 3 prime to 5 prime, means it will be anti-parallel. That is the meaning of anti-parallel. These strands are complementary and anti-parallel to each other. They are spirally coiled. These 2 strands will spirally coil each other, and each polynucleotide strand is made up of 1000 nucleotides. Nucleotides are structural unit of DNA. So this DNA is the smallest unit of DNA is a nucleotide. So it is the structural unit. The structure of a DNA is formed by the nucleotides itself. This is a very important point you need to keep in mind. That is, nucleotides are the structural units of DNA. Each nucleotide is composed of 3 components. What are the 3 components of which these nucleotides are made up of? Each nucleotide is composed of 3 components, namely a nitrogenous base, a molecule of pentose sugar, that is deoxyribose form, and a molecule of phosphate. Let's see the structure. You can see here the structure of a nucleotide. You will find here this pentose sugar, phosphate group, and this is the nitrogenous base, which is represented by NB. You can see here the two strands which are anti-parallel to each other, isn't it? You can see the structure itself. First of all, it will start from a phosphate group, pentose sugar, and a nitrogenous base. Then again, the next nitrogenous base, pentose sugar, and the phosphate group. These two nitrogenous bases will join with each other with a hydrogen bond. So you can see here like that two nucleotides I have made. This is a part of DNA molecule. This is a part of a DNA molecule, and here you will find double helical structure. This is a coil structure of DNA, where you will find the two strands which are spirally coiled with each other, where you can find the different nitrogenous bases. That is A stands for adenine, thymine, T stands for thymine, G stands for guanine, and C stands for cytosine. So this is the structure. You will also find that in between adenine and thymine, the number of hydrogen bond which is present is 2, whereas between G and C, that is guanine and cytosine, the number of hydrogen bond which is present is 3. So in the same way it will be, the nitrogenous bases will be arranged. These are the nitrogenous bases which is represented here by Cg. This nitrogenous bases CGTA, and this phosphate sugar it will be present this side. Now coming to the nitrogenous bases, there are 4 types of nitrogenous bases found in DNA. What are those 4 types of nitrogenous bases? The first one is adenine, A, represented by alphabet A. Next one is guanine, which is represented by capital G or alphabet G. Furthermore, moving ahead, the next type of nitrogenous base is thymine, which is represented by alphabet capital T. And the next one is cytosine. The last one is cytosine, which is represented by C. Now, this adenine is complementary to thymine. So, A will always pair with T, means adenine will always pair with thymine. Whereas, guanine is complementary to cytosine, and it will make a pair with C, means G will form its pair with C. The nitrogenous bases, that is adenine, and combines with thymine using 2 hydrogen bonds, and guanine with cytosine using 3 hydrogen bonds. The arrangement of sugar, that is phosphate sugar, and nitrogenous base gives us spiral ladder-like shape. If you I have shown you in the diagram, it forms a spiral ladder-like structure. Spiral ladder-like shape to the DNA molecule. This is why the structure of DNA, double-stranded DNA, is termed as a double helical structure of DNA. The next topic that we will see is formation of new DNA molecule. How does the formation of this new DNA molecule takes place? Formation of new DNA molecule. It is also called as duplication or replication of DNA. Here you will find a double helix DNA. You can see the two helix, the two strands of a DNA, which is represented by 2 colors, white and blue. Okay, so what actually happens as the replication or duplication during the process, as it will start, the strand from here, it will open. It will open. And new strands will start, new nucleotides will start to accumulate. You can see here with green color. It is opening, unwinding from one end. At the same time, new nucleotides are taking place and forming complementary bases. Okay? And it will move ahead in this direction. Strands open at one end and forms new strand. You can see here new strands are being formed. The uncoiling process is taking place, and at last we will find two daughter chromosomes are present. They are formed. This takes place during S phase of interphase. So this will take place during the S phase of the chromosome. What is the S phase of the S phase in interphase? That is the synthetic phase or the synthesis phase of the cell cycle. During this process, the hydrogen bond of two strands of coiled DNA helix, they will break. You can see here it will break. It is breaking from here and starts to uncoil and separate and continue forward along the whole length. Along the whole length, it will start to continue to uncoil and separate and continue forward along the whole length of DNA. New nucleotides join the exposed nitrogenous bases. You can see here new nucleotides are joining and forming a new strand, and it will this thing will move on in the same direction. This will form this forms two new polynucleotides strands complementary to the two parental strands. So actually this, if you see, this is the parental strand, this blue, and represented by white strand, and new nucleotides are attaching and forming the daughter strand. Okay, so at last we will find two daughter strands are formed. It forms two new polynucleotide strands. You can see here two new polynucleotide strands are formed, complementary to the two parallel strands. At the end, two daughter molecules are formed. You can see here at the end, after completion of this thing, the two daughter molecules are formed, or the replicates are formed, and in this way the replication completes. The next topic that we will be seeing is gene. Okay, these genes are located on the chromosome only. These are specific sequences, nucleotide sequence, which are found on the chromosome itself. So what is a gene? It is the unit of genetics or heredity. It passes traits from one generation to the other generation. It is just because of the gene that we resemble to our parents, that we resemble to our ancestors. Okay, so it happens due to the transfer of genes from the parents to the offspring and so on. It means it is helpful in the transfer of phenotypic or physical characteristic from one generation to the other generation. So gene is very important. The traits are determined by types of proteins formed in the cell. So different individuals are having different traits, different characteristics. So these are determined by the type of proteins which are formed after encoding the particular genome. The protein expresses itself in the form of specific character of a body. The protein which is produced, it will be expressed in a specific type character of a body by enacting as an enzyme. That protein either they may enact as an enzyme or they may form a structural component. What is the definition of gene? A gene is a segment of DNA having specific sequence of nucleotides that encodes a particular protein. Again, I'll be telling you, a gene is a segment of DNA having specific sequence of nucleotides that encodes a particular protein. So this is a very important definition. What is the function of this chromosome? As we all know, chromosomes are the genetic vehicles carrying genes from parents to offspring. From the parents to the offspring, all the traits are transferred by this chromosome itself. Actually, the chromosomes are having genes that are responsible for the transfer of traits. They control the metabolism and other activities of cell by directing synthesis of required protein. So they will direct the type of protein which is required by the body. It directs various life processes like cell growth, cell division, and cell repair. They guide development and cell differentiation. The development and the cell differentiation process is guided by the chromosome itself. Chromosome X and Y determines, there are two types of chromosomes basically, male and female, in male and female, and that only determines the sex of an organism. Means chromosome X and chromosome Y, they determine the sex of any organism. So we will be seeing the same thing again, whatever I have taught you through a PowerPoint presentation now. Starting. Structure of chromosomes. Chromosomes are small, rod-shaped, darkly stained bodies visible during metaphase and anaphase of nuclear division. The genetic material in the resting phase, that is non-dividing nucleus, is in the form of very long, extremely thin fibers called chromatin fibers, which will form the chromatin net. This network is called chromatin net. When a cell enters cell division, the chromatin fibers starts to condense. They attain into thick, dark stained bodies called chromosomes. Chromosomes are formed of nucleoproteins. What are nucleoproteins? That is nucleic acid, which is DNA, and basic protein called histones. Chromosomes are called vehicles of genetic material. Why are they called as vehicles of genetic material? As they are transferred from parents to offspring, means from the parents to their children. That is from one generation to the other generation during cell division. Discovery of chromosome. Chromosomes were first observed by Hofmeister in the year 1848 in the nuclei of pollen mother cell of Tradescantia. I had shown you the Tradescantia plant also today. The term chromosome was used by Valder in the year 1888. Next is chromosome number. Number of chromosome is constant in all the cells of an individual and individuals of a particular species. Means all the individuals of a species belonging to one species, they will have a single number of, it's the same chromosomal number. Whereas organisms belonging to different species will have different chromosome number. Therefore, number of chromosome is helpful in identification of species. Let us see some examples of number of chromosomes in plant and animals. You can see here the examples which are given. These are the example of some plants and animals. The first one is onion, that is Allium cepa, with number of chromosome in diploid form is 16. Second, rice, that is Oryza sativa. number of chromosome is 24. The very next is wheat, Triticum vulgare, number of chromosome is 42. The next example is man, that is Homo sapiens, the scientific name, with chromosomal number 46. And last example is cat, that is Felis domesticus, and the diploid number of chromosome is 38. Diploid and haploid number of chromosomes. First of all, we'll be knowing what is diploid number. Each cell of body has two sets of chromosomes. One set comes from sperm, that is a male gamete, and the other set from ovum, that is female gamete. So it is represented, this diploid number is represented by 2n. There are two ways, 2 small n or 2 capital N. Both the ways are correct representation of diploid number. Next is haploid number. These are found in the germ cells or gametes of diploid organisms. They contain only one set of chromosomes. Since they are present only in the germ cells, they will contain only one set of chromosomes. They are represented by a small n or a capital N. Structure of chromosomes. Each chromosome appears in its condensed form during the start of cell division. It consists of two chromatids joined at some narrow constricted portion, as it was shown in the diagram. It is marked as centromere. I had told you its name is centromere also. It can be called as a kinetochore or primary constriction region. The region of chromosome on either side of centromere is called arm. What is centromere? It is a point where spindle fibers are attached during cell division. What is the function of centromere? It determines the shape of chromosomes. It provides attachment for spindle fibers. It helps in the movement of chromosomes during cell division. Now let us see what are the different types of chromosomes. Classification of chromosome is based on the position of centromere. The first type is telocentric. These are rod-shaped chromosomes with centromere occupying a terminal position. They possess only one arm. The next type is acrocentric. These are rod-shaped chromosomes with subterminal centromere. Means slightly just before the ending point. One arm is very short, whereas the other arm is very long. Other arm is long, and the first arm is very short, and the other one is long. The next type is submetacentric. These are L-shaped chromosomes, and the centromere is slightly away from the midpoint, due to which an alphabet L-shaped is attained. The two arms of the submetacentric chromosome are unequal. Next type is metacentric chromosomes. These are V-shaped chromosomes. They attain in English alphabet V, the shape of English alphabet V. The centromere here will lie in the middle of with two equal arms. We will find the centromere situated in the center, and it will have two equal arms. Here you can see the structure of the telocentric, acrocentric, submetacentric and metacentric chromosome, where you will find in telocentric the centromere is terminally located. In acrocentric, you will find it is not terminally but subterminally it is located. In the metacentric, you will find one, it has attained an L shape. One arm is short and the other arm is long. Metacentric, you will find the centromere located in the center and it has attained a V shape, English alphabet V shape. This types of chromosomes is based on the position of centromere. Next is fine structure of chromosomes. Chromatin material condenses to form chromosomes. Chromatin is formed of two substances. What are the two substances of which this chromatin material is made up of? That is deoxyribonucleic acid, short form is DNA. Basic proteins, that is called histones. So these are the two things, that is DNA and histones, of which this chromatin material is made up of. DNA forms 40% of the chromatin material. Histone forms the remaining 60%. Altogether 100%, means 60% is histones, 40% is DNA. Both this DNA and histone, they form a complex structure. The structure which they will form, here histones form a central or small core particle, small core particle, and DNA is found wrapping around it. This forms a structure called nucleosomes. Each nucleosome has one core particle formed of 8 molecules of histones. DNA makes 2.5 coils around it. Chromatin fiber has numerous nucleosomes which are arranged like beads on a string. I have given you the example of a pearl necklace. From that you can understand it forms the numerous nucleosomes which are arranged, they are arranged like beads on a string. You can see here the structure of a nucleosome, which is showing histone proteins. Numbering is shown. Number 1 is histone protein, number 2 is DNA strand, and 3 is linker histone. Structure of DNA. DNA is a macromolecule formed of 2 polynucleotide strands. These strands are complementary and anti-parallel to each other. I have told you the meaning of anti-parallel. If one strand will start from 5' to 3', the other will start from 3' and end with 5'. That is the meaning, that they are anti-parallel. They are found spirally coiled with each other. Each polynucleotide strand is made up of thousands of nucleotides. Nucleotides are the structural units of DNA. Each nucleotide is composed of 3 components. What are the 3 components? A nitrogenous base, a molecule of pentose sugar, that is deoxyribose sugar, and a molecule of phosphate. There are 4 types of nitrogenous bases which are found in DNA. What are those 4 types? Adenine, represented by alphabet A, guanine, represented by G, thymine, represented by T, and cytosine, represented by C. You can see here the structure. a basic structure of a nucleotide, which is represented in A, where you will find a phosphate group, sugar and base, and two parallel strands of a part of DNA. You can see here the two strands which are anti-parallel to each other, and in the center you will find the nitrogenous bases are present, which are joined by hydrogen bonds. Adenine is complementary to thymine. So adenine will always pair with thymine, that is A will always pair with T. Guanine is complementary to cytosine. So G will make pair with C, means guanine will make pair with cytosine. The nitrogenous base, that is adenine, combines with thymine using two hydrogen bonds, means it will have two hydrogen bonds between them. And guanine will combine with cytosine using three hydrogen bonds. The arrangement of sugar, phosphate and nitrogenous base gives a special ladder-like shape to the DNA molecule. You can see here the spirally coiled DNA molecule, a ladder-like structure. You can see here this ladder-like structure, the hydrogen forms, you can see it forms the rung of a ladder. You can see three hydrogen bonds which are present between cytosine and guanine, two hydrogen bonds which are present between adenine and thymine. This is one complete turn of DNA spiral. This is why the structure is termed as double helix and forms a double helical structure of DNA. Formation of new DNA molecule. How does the formation or duplication or replication of DNA takes place? It takes place during the synthetic phase of interphase of the cell cycle. During this process, the hydrogen bonds of the two strands of coiled DNA helix break apart. Strands starts to uncoil, separates and continue forward along the whole length of DNA. New nucleotides join to the exposed nitrogenous bases. Forms two new polynucleotide strands. Thus two new polynucleotide strands are formed. These two polynucleotide strands are complementary to each other, complementary to the two parental strands. They will complement to the two parental strand that has separated from each other due to uncoiling. At the end, two daughter molecules are formed and thus replication completes. You can see here it has been shown with the help of diagram. In the first, that is A, you will find a double helix of DNA. It starts to uncoil. As the old strand will uncoil, new strand will start to develop by the deposition of new nucleotides, and it will move forward. Strands open up at one end, and as a result, two new strands are produced. At last, you will find each new DNA contains one strand and one old strand and one new strand. Next is gene. It is the unit of genetics or heredity. It passes traits from one generation to the other generation during sexual reproduction. The traits are determined by the type of proteins formed in the cell. The protein expresses itself in the form of specific character of the body. This protein either enacts, means it will act like an enzyme to do its function, or it forms a structural component. Definition of gene. So this is important. Definition of gene. You can also note it down in your copies. A gene is a segment of DNA having specific sequence of nucleotides that encodes a particular protein. Again, I'll read. A gene is a segment of DNA having specific sequence of nucleotides that encodes a particular protein. Let's see the function of chromosomes. Chromosomes are the genetic vehicles carrying genes from parents to the offspring. Chromosomes control the metabolism and other activities of cell by directing synthesis of required protein. They direct various life processes like cell growth, cell division, and cell repair. They guide development and cell differentiation. Chromosome X and chromosome Y determines the sex of an organism, whether it is male or female. As we know, in males the chromosomes are X and Y, and in females it is X and X. So this was all about the chapter today, and you have understood the chapter. Upon your understanding, there are some questions that you are supposed to do in your copy. You can see here the questions. You need to write the answers of the questions which are given here. I will read out the questions. First is, what is a chromosome? Second question, who first observed chromosome and in which organism? I had shown you the organism also today. Name the bonds. Third question, name the bonds which joins the complementary nitrogenous bases of polynucleotide chains of DNA. Fourth question, give the functions of centromere. Means you have to enumerate the function of centromere. Give the basic rule of nitrogenous base pairing. There are certain rules that nitrogenous base has to follow, that you have to write down. So these are the 5 questions. The answers are very simple. It has been told to you during my explanation. So you have to write the answers of these questions. So that is all for the day. Goodbye.